So this is the talk before the August summer break, our last talk, and I hope a lot of people will still be joining us. It's a great pleasure to have Chaitanya Atale actually joining us for presenting his work on the quantification of cell shape and intracellular flows based on DIC contrast, differential interference contrast. So just introduce our speaker a little bit more about also his work. So he has been working on label-free imaging, and because label-free imaging has undergone quite a revitalization with improvements of algorithms and computing power. But of course it's combined with fluorescence tagging, and I think anyone using confocal microscope, I always teach they should not forget to use DIC or phase contrast as well, because it's actually a fantastic complementary technique at least. But in his talk, Chaitanya will discuss two aspects of his work about differential interference contrast applications to E. coli imaging, as well as also to C. elegans imaging, and there in particular the flow to spindle oscillations. I mean, so just to introduce Chaitanya personally, he studied zoology at Pune University in India, where he is currently speaking to us from now. He went to do a PhD at the University of Heidelberg, Germany, with Roland Ailes in modeling and measuring intracellular diffusion. And his first postdoc was at the Mass General Hospital in Boston. And the second postdoc, he returned, I think maybe to Heidelberg, working with Eric Casenti on microtubule regulation. So since 2009, he's been working in Pune and looking at the role of cytoskeleton and cell shape by a combination of computer simulations, in vitro reconstitution, and microscopy and image analysis. So it's a pleasure also to point out Chaitanya actually proposed this talk and came forward from the website, introduced himself to us. So anybody in the audience, if you would like to kind of do the same, we're very open to people joining Imaging One World and seeing your work here presented. We don't want to just kind of find people. And please, any PhD student, postdoc, if you have imaging techniques data like to present, please feel free to kind of contact us. So please, can you tell us about your work? I'm excited.
Thank you very much. Thank you very much for the kind introduction, Stephanie. And I see some old friends also in the audience. Nice. Francesco, hi. And I also, this is maybe a little unusual, because I guess my only connection with the Royal Microscopic Society so far has been, in fact, a paper we wrote up on some low-cost imaging, and again, based on a bit of image analysis. But I'm not going to be talking about that today. I'm really going to try and focus a little bit on something that we've been doing over, in fact, the last 11 years in the lab when I started, on indeed cell shape quantification and intracellular flows. And I think everyone in this audience is familiar with it, but I'm just going to, for the sake of completeness perhaps, go over a few things of what I crudely see as the basic background to DIC imaging and the purpose in image analysis. So this is really going to be an image analysis talk focusing on gradients detection, how we applied it coli, and in a way, perhaps even naively applied it. And then we sort of improved on it a little bit with some Gaussian filtering, modified Gaussian filtering, and use that to infer intracellular flows. And I'll sort of try to wrap it up like that. And so, of course, it's always nice to go back to the basics. And I must say that the principle of DIC, as Stephanie very nicely pointed out, much as it is an old technique and it's label-free, and in this day and age, at least in biological microscopy with fluorescence taking us from strength to strength, it sometimes becomes easy to forget about DIC because it doesn't, of course, have molecular specificity in an intracellular milieu where you have multiple molecules also playing a role. But at the same time, this basic principle of polarized light passing through a prism giving you two components which then are differentially sheared in the specimen, recombined in the prism, and passed through the analyzer to give you a differential interference patterns of constructive and destructive interference, resulting in contrast with absolutely no labeling, is powerful in itself, right? And I'll be slightly honest here. One of the reasons I started doing this is simply because when I arrived in 2009 to set up my lab and I asked for a microscope and it didn't come for 3 years, the only microscope there was one with a DIC and a standard fluorescence lamp. So well, I mean, necessity is the mother of invention, and that's how we went about it. But of course, I mean, it forced me also to go back and look at the background to some of the things that have been done, and so-called the Olympics of DIC in terms of what all has been achieved, pushing in some ways, in my view, the method to its limits in terms of ability to resolve and ability to also look inside a cell where things are naturally crowded. And this idea of crowding is, of course, evident to anyone who has done a rough calculation of the number of proteins in a cell and the spacing. I mean, if you look at an E. coli cell with 10 to the power 6 molecules, you expect approximately a spacing of a few nanometers, assuming point assumptions. I mean, this is, given all of this, we know it's crowded. So to me, it was almost a surprise then to see in very early work in 1980s, I'm sorry, not very early, but relatively early work in the 80s of Ted Salmon and Claire Waterman Storer, showing that they could in fact resolve single microtubule filaments inside the leading edge of a newt epithelial cell. And this is sort of impressive because, of course, this was at a time when digital microscopy and cameras were not as advanced as they are today. And yet they could in fact use some maybe primitive methods of image analysis. And in the second paper by Walker in '88, in fact, an even older paper, they used this to not just detect filaments, to view them, but also used what we would today call interactive or manual segmentation method to detect the tip and quantify dynamic instability of microtubules. And I know that not everyone here is familiar with microtubules, and I did not say much about it. So please do stop me if you think that I am rushing through this. But suffice to say, microtubules are one of the major cytoskeletal elements inside a cell. They form these canonical 13 plus minus something protofilament structures in a cylindrical geometry with a helical rise. And these structures, because perhaps of their regularity, are very good samples for this kind of label-free imaging, which is why this lower panel that you see from Walker's paper shows up extremely sharp contrast in a pure system. So the lower panel is, of course, in vitro. In other words, purified microtubules and a purified axoneme, which was used to nucleate the microtubules. But on top of that, going back to the in vivo situation, people have also been able to quantify flows, and these have again been done by following the kind of granule motion. So maybe I just want to play this movie again because it's so beautiful in my view. But more importantly, if you notice on the top, there are also little intracellular vesicles, large structures that are also moving around in what appears to be a sort of random fashion. Now, at the same time, the fact that cytoskeletal systems, and in this case now the actomyosin system, is capable of driving collective, cohesive, directed transport can result in intracellular flows. And the classic example for this is, in fact, the plant cell, Cara, which has one of the largest single cells. In fact, the so-called internodal component between these two structures that I'm going to crudely call leaf-like objects is just one cell, and it's a few millimeters in length. And at this scale, to generate systematic flows is impressive, and maybe for another occasion, something related to the collective action of molecular motors and the cytoskeleton, in this case, actin and myosin. But it also seems to have a physiological role. So studying intracellular flows is not just a sort of nice thing to look at because they're moving. I mean, anything that moves, we enjoy. I as a microscopist, I think it's the most joyful thing. But there's more to it, because apparently when the group of Ito and Tominaga introduced mutant motors, which had, in fact, hybrid motors, which had wild type or faster than the wild type or slower than the wild type speeds of the myosin, they could in fact see that the number of cell divisions remained the same, but the total effect on the plant, as you can see in those two images in the lower panel, lower left panel, was that high-speed motors led to bigger plants and low-speed motor led to slower plants, smaller plants. And this came out of the regulation of the intracellular flow rates. And this is slightly odd in a way, which also suggests we don't completely understand it. And so in a way, understanding this in a clearer sense, getting numbers out of it, and perhaps even being able to develop a model for it would really be of some use in our view. And for obvious reasons, when you are looking at flow, the first thing that comes to mind is probably a method which, from what I understand, comes from engineering physics, engineering called particle image velocimetry. And this example that I've pulled up here from Ganguly and Company's paper in the PNAS suggests that they used a combination of passive tracers, which were endogenous granular particles that show autofluorescence when imaged, and the fact that microtubules were also labeled in the posterior chamber of the oocyte of Drosophila, demonstrated that they could see flows. And this was illustrated by the fact that when they then analyzed, so left to right in the rightmost panel is the time series, the two snapshots in time effectively. When they analyzed in a grid of 3x3 interrogation, the time gradients or the particle movements in terms of direction and magnitude, they could generate this vector field map. And this vector field map so beautifully illustrates the fact that there are global flows happening. And of course, this method is nice. It gives you some, excuse me, gives us insight into the process. It can also be used for interrogating the effect of genetic modifications. But at the same time, we think that this kind of a particle velocimetry approach, which in this case now has also been used in the case of C. elegans embryos, the nematode, the classic genetic model organism, has probably some limitations in our view. And the limitation we are most interested in is the fact that what happens to the fate of the individual objects. The point that you obviously, I hope you can make out here, is that the power of the PIV approach is indeed to give us some kind of mean field approximation of flows. And it of course provides a spatial information. But in contrast, single particle tracking, which would do both of these things effectively, does also allow us to follow the so-called history or the lineage of those particles. And in biology, as often, weird things happen to objects. So vesicles may divide, they may move, they may fuse. And addressing these kind of so-called morphological characteristics is the real purpose for us of pushing the limits of what we can do with single particle tracking in order to get similar ideas of cytoplasmic flows that people have shown already using differential interference contrast microscopy with endogenous particles like over here, yolk granules in C. elegans. So I hope I will come to that and maybe discuss a little bit of how we went about doing it. But for the first part, I want to discuss a little bit about what we did in terms of a very simple, maybe low-level approach to detecting image gradients and using that to perform cell size analysis with bacterial cells, in this case, E. coli. And much of it was motivated by the fact that we have a sense of wonder when we look at natural variability. And I like to think of it as the long and short of the tail, not just the story, but also because the distribution of cells in a population, even when synchronized, is not normal. It's not a Gaussian. So we in fact found that cell sizes seem to have a long tail distribution. And when we compared what we had in the freezer and grew up, the so-called lab strain, which we can sometimes in literature refer to as wild type, E. coli MG1655, compared to the lab workhorse for cloning, DH5 alpha, we were struck by how different these populations are. And this is taken from a midlog culture as a sample to represent the fact that there is a big difference. There are 2-3 questions that struck us at this point. Why is there a difference? Why are the cells not uniform in E. coli MJ1655? Is it just simply because we're looking at a mixed population which could be at different cell cycle stages? Or is there something else going on? And can we then use that to explain why these DH5 alpha are enormous? And by enormous, I mean approximately 20 to 30 micrometers in length. Oddly enough, also by eye, you can tell probably their widths are not very different. And this is sort of an aside, and I'm not going to dwell on it today, but it's something that we've been now beginning to get a little bit of an understanding of. And so the experimental design was such that we grew our cells at different temperatures. And part of the reason was because when I saw this and I went to a microbiologist and I asked them, why do you think cells get longer? And they said, oh, well, temperature affects them. It was when we grow them at lower temperatures, they become longer. And when we grow them at higher temperatures, they become shorter. So obviously, this is a very qualitative statement. And I went back to the old papers, 1960s, 70s. It's a great pleasure to read those. Either way, we thought temperature may be a good modulator. So we went ahead and did the experiment. It's a fairly straightforward experiment to do. There was a master's student in my lab and she was more than enthusiastic to grow cells. So we did that and we got these nice looking growth curves over a range of what we may call roughly permissive temperatures, 22 to 42 degrees Celsius. And these curves, as expected, showed that lower the temperature, the lower the growth rate. This is what you see in panel A. Higher the growth rate, the steeper the curve looks. 42 degrees grows at a rate that may be slightly faster or similar to 37, but also saturates at a lower rate. In other words, the carrying capacity is a bit lower because it's not an optimal temperature for the growth of those bacteria. So when we did this and we estimated some sort of doubling times from those, we got a systematic decrease in doubling time or increase in growth rate as you expect. And the microscopy that we did was basically sampling. We were sampling from bulk populations. Again, in a way, perhaps these are statistical representations of what we see in a population. They could be in multiple cell cycle stages. And what we observed then, of course, was that lower the temperature to the eye, the cells seemed to be shorter. Higher the temperature, the longer they seemed to become. Nice qualitative observation, but what do the quantifications look like? In order to do that, we decided to use something that we could tell by eye, and this is something that is very well known in DIC imaging, that you get a pseudo 3D representation, right? It's an apparent three-dimensional structure seems to emerge from such images, which arises due to some form of an image gradient formed at edges. Now, because E. coli cells, rod-shaped cells are elongated along one axis, it's sort of apparent that maybe capturing this shear gradient would perhaps give us some information about the cell size, because at that time, all we wanted to know was length, because it looked from literature and from a lot of other sources that in E. coli at least, width is not so much, does not vary so much with conditions, but probably it is the length. And so what we did was wrote down some simple code to do a pixel-wise gradient analysis, where the neighborhood of this gradient is defined by this n term, and this n term decides the size of the filter. So it's nothing but basically comparing a filter of a certain size with a pixel and running it through the image. And not knowing the direction of the shear, we calculated an X and a Y. And now one could have, of course, done this more elegantly, but this was, as I said, a naive first approximation. And when we did that, we got a gradient image, which we binarized by thresholding based on a gradient, a threshold, which was interactively set, which matched seemingly most of our images taken under the same conditions of illumination. This, of course, gave us then by multiplying the binary thresholded X and Y gradient images to one final gradient image, which we see in the top part over here in the rightmost panel. And then we cleaned up the image to get rid of small specks, which seemed to have also appeared, to get us something like length. And so this then, of course, led us to think, well, are our statistics correct, right? Because, I mean, this is perhaps maybe a very simple and crude way So the first thing we did was we counted the frequency of false positives, false negatives, true positives, and true negatives. And the algorithms 1, 2, 3, 3 is corresponding to our method of this so-called gradient detection. 1 and 2 were a standard method of canny edge detection and simple filtering, Gaussian filtering. Now, when we compared these, it looked like we probably had something, but when we needed a ground truth. And the ground truth that we used was so-called interactive or manual measurement, averaged over 3 individuals in the lab, and they gave us the numbers. We cross-correlated that they'd not done something strange with their data, and looked to see whether the lengths that were analyzed by the algorithms, by the method, were disproportionately larger or smaller than those analyzed by the algorithm. And our method and the simple gradient filtering gave us comparable numbers. And at the same time, the gradient detection seemed to catch a higher proportion of what we consider to be the ideal number of cells. So we thought, okay, we're probably not too far off from what it ought to be. What about any other method of labeling? Because all of this is sort of manual, is a bit tricky. So we also labeled the cells with a membrane dye, FM464, and used standard Otsu thresholding to get cell boundaries, measured the lengths from those using inbuilt methods in ImageJ, and then compared those values. And it looked to us like our lengths detected were somewhat more closer to the manual detection from DIC gradient methods as compared to membranes, but all within an error bar. So we could not really find a difference. So in other words, we thought they were very similar to each other. And finally, we looked at whether our filter size makes a difference. And it looked like, at least for a small range between 2 and 5 pixels, for this resolution, this was a 63x magnification on a Zeiss epifluorescence microscope. It did not make much of a difference. So the last thing we needed to deal with was the fact that occasionally in these fields of view, our method relied on a skeletonization approach. So once we had detected gradients, we often got branched structures, and these branched structures we wanted to resolve. So we used basically an iterative method to go to branch points. Branch points were then defined along a skeleton, 1 pixel wide skeleton, as those skeleton pixels that had more than 2 neighbors. And at that point, we used an angle minimization to find the minimum of the branches. So this sort of allowed us to prune things, and basically the assumption, of course, being that cells are less likely to form these V shapes and more likely to be elongated. Now, this is a working assumption. More or less it applies. If we were looking at mutants, we may have missed some things, but we were not. So for the moment, it worked reasonably well. We also got less data loss. In other words, that we wouldn't have had to discard that data. And so when we then did that, we got these nice length distributions. And to answer my original question, does temperature affect cell length? It looks like in the early and late log, mid log and early and late log phases, the rapid growth phases, the distribution of cell lengths is wider at higher temperatures and tighter or narrower at lower temperatures. And the stationary phase distributions look almost like each other. And part of it is probably related to something that has a connection to growth rate. So in order to address this, we thought we should probably modulate growth rate by something less general, less physical than just temperature. Just a small note over here to say that if you look at the fit values to these log normal fits to the frequency distributions indicated by the mu values, as the temperature increases in mid-log phases, early and late log phases, the lengths of those cells on an average increases. The tails also become slightly fatter. Yeah. At the same time, when we then used different sugar sources with minimal media, which are essentially some kind of defined growth media where we can provide one single carbon source in terms of a sugar, either acetate, succinate, or glucose. And for those of you who are familiar with bacteriology, you remember that glucose leads to faster metabolism and faster growth, simply because it's a preferred sugar in the metabolic pathway. And as perhaps expected, the increased growth resulting from such sugar supplements led to these growth curves that looked steeper and steeper. So going from the purple through the dark red, deep red, to the blue onwards to red, and also resulted for LB, which was this sort of undefined but complex medium, to the widest spread in mid-log phases, and a narrower and narrower spread as we went to a so-called more nutrient poor medium. And all this came straight out of our cell length analysis. So we could sort of answer perhaps the question that we began asking, which is that does the growth rate affect cell size? But we of course wanted to go a little further, and we went back to our extremely elongated 40 micron cells and did the same analysis. But this time, because we had the geometric outline, the skeleton of the cell in place, we could use correlative microscopy. So we had to align our two fields of view. We had a DAPI stain for the DNA and a DIC image, which was segmented to give us the so-called cell skeleton or central line. Along that, we had a profile, and that gave us an opportunity to segment out how many discrete points of DNA are present. And these, in bacteria, as you remember, perhaps these are membrane, they lack organelles or bona fide membrane intracellular structures. The DNA is naked in the sense that it is not bounded by a nucleus. At the same time, it seems to still compact into a tight ball, at least in interphase, and during replication segregate. And these structures are then referred to as nucleoids. So we counted the number of nucleoids, and to our surprise, we found that as the number of nucleoids increase, the cell length increases in a way in these DH5 alpha cells, these lab workhorse strains, which has a very nice slope of approximately 2 micrometers per nucleoid. And for those of you who remember sort of standard numbers, and if you don't, I am more than happy to tell you, the typical, canonical textbook picture of E. coli is that the birth length of E. coli is 2 microns. So a slope of 2 microns per nucleoid suggests that there is a failure of division, but not of cell growth and not of DNA replication. And so this was a very nice little insight that came simply out of DIC image analysis, some morphometry, and correlative analysis with a fluorescence channel and peak finding algorithm. So this is a method that we've described, and we put the code out online, and anyone who wants to see it can come to our website, or we can also share it if you drop an email. And this sort of led us to this idea that maybe this method could be used a little bit more widely. But a student came over to my lab who one day showed up and basically told me that this method is not very efficient. The code is very poorly written. I agree. So we sat and tried to write a better piece of code, and that's what led us to a little bit of tweaking of this approach. Before I go any further, are there any questions perhaps for this part? I'm going to change gears a little bit. So.
I don't see any questions in the chat, Laura.
Oh, okay. I'm sorry.
They're not in there. So I'll just put a message in there so people can type them into the chat. Okay.
Yes. Okay. Yes. I see.
Yeah, we do the Mentimeter at the end, and then we have the questions. So.
Yes. Voila. Okay. No
so we have about, I think it's about 10 minutes because we like to keep 15 minutes. Is okay for the second part?
Yes, that's perfect.
So then we have 15 minutes for the questions. So thanks.
Super.
Yeah, super nice.
Yeah, great.
Enjoying this.
Yeah, okay. So I shall continue. Okay. As I said, the problem we began to tackle was slightly bigger cells. As you see, these are approximately 50 micrometers across and 20 microns in the short axis. And this varies. And this is the nematode, the famous genetic model organism, Caenorhabditis elegans N2. There's enough been written about it. Suffice to say, this is the start of life for the C. elegans. This is the one-celled embryonic stage. And in this one-celled embryonic stage, in C. elegans at least, the spindle that is formed after fertilization appears to undergo some oscillations in the anterior-posterior direction. And so when the so anterior and posterior is decided at the time of fertilization. So in fact, what is anterior and posterior in one-celled embryo, which you cannot see morphologically distinguishable by any means, eventually becomes the head and the tail. So this makes C. elegans such a beautiful developmental model organism. And those of you who study it, of course, are very familiar, so I won't belabor the point. But these oscillations have fascinated a lot of people in the cellular biophysics field, because obviously, why does something oscillate? I mean, it's a little unusual. On top of which, along with that, why it oscillates, it's also an interesting question, what else moves along with it? And so here, what had been tracked, both using manual or interactive approaches as well as analysis by the lab of Marie Delattre, who is now a collaborator, was the clear region, the DIC clear region in these embryos. And as I was informed, in fact, by Marie, one of the reasons for the choice of C. elegans or rather, one of the choices that drove one of the reasons that drove the choice of C. elegans as a model organism was also this beautiful optical transparency of its early embryonic stages. What you are in fact looking at, the granular structure, these are something I'm going to come to, but are yolk granules as far as we're aware. And these yolk granules are birefringent. They give beautiful contrast. And when you zoom in much further into them, you in fact see that they have this kind of classic DIC dark shadow and a bright part. Now, this is great for the eye. Our brain is great for it. But can we even bother trying to segment this? So one of the things we tried to do was build a sort of scaled Gaussian filter, which we call scaling of Gaussian, where we take a Gaussian and subtract it with a scaling factor, which we have decided based on the image statistics, and get a Gaussian profile, a filter profile, which has both positive and negative parts. Now, this is kind of trivial. It's basically like subtracting a constant from it. But to our minds, because of the way we structured the filter, we could deal with both predominantly bright objects and predominantly dark objects by simply swapping parameters. And so this sort of helped us, because when we then looked at the ROIs of these granules, this is a zoomed in view from the nematode embryo, then the filtering almost gave us something that looked like what you might in fact call a fluorescence image in the sense of the grayscales. This allowed us to very simply do a standard or two kind of thresholding and find centroids. Now, our aim was not so much to determine the morphology, but the identity of these granules. And that's really where we went with. And so now that we had these beautiful granules fairly well segmented, we went back and checked that our statistics were reasonable. We tried to do some sort of similar sort of ground truth analysis. Our ground truth again became manual by eye detection, and comparing the filter that we were using, this kind of scaled Gaussian, we tried to estimate what is the error in positional detection as compared to standard algorithms that one would have normally thought of when you're trying to segment in a filter, in order to segment DIC images with this kind of granular structure. And the difference of Gaussians is the most natural one to think of. We also looked at the Gaussian, the blue, and an inverse Laplacian of Gaussian, which have all been reported to work fairly well on this kind of data set. And it looked like the positional detection error was marginally lower on an average. These are the light green bars in this plot here in F. But at the same time, the spreads were much tighter, which also meant that it may have been a more reliable measure for us. The mean and median values also looked sort of around a pixel. Now, we would, of course, love to improve the pixel detection error. But as you see in this very, as we increased the noise level, so the x-axis is the variance of the noise that we've filtered in, this becomes really hard to do. Now, we, of course, always argue that in order to do a decent job with segmentation, you would rather not work with noisy images. So indeed, within the limits of V is equal to 0 is our zero noise image. The noise is anything that came intrinsically from microscopy and the camera setup. We hope that we are dealing with, at the very most, 1 pixel detection error. And at 100x magnification, this is something in the range of a few hundreds of nanometers in terms of absolute pixel micrometers. So spatial units. But so we are not sub-pixel, which we would love to do, but we are at least sub-micrometer. And given this, we then threw in a single particle tracking method, which then was based on a minimal distance and branch elimination approach algorithm, which was a standard code that we've taken from Howard Berg's lab and modified with all our add-on segmentations. And to our surprise, we actually could see fairly decent over almost a few minutes timescales movements that beautifully correlate with the spindle oscillation. So if you remember, I mentioned in my motivation for this problem, the fact that the spindle itself is moving. I mean, it was a question to us whether the granules follow it passively or are they stuck because of some other intracellular structures. And it looks, at least to the eye, that our tracking seems to capture them. We can follow it, even though there may be Z movement in terms of granules going in or out of the plane. And we could also then quantify this. And when we did this quantification, we did a little bit of alignment of these trajectories. We basically measured the displacement in Y and X from the respective origins of those individual granules. We got a large data set, in fact, and did a fast Fourier transform to get out, after some smoothing, to get out some characteristic frequencies. The frequencies we got were around 40 millihertz. And these, to our surprise, actually correspond to what people have reported for spindle oscillations. So this is the dominant frequency as far as we can see. I think some of you may notice there's a low frequency peak at the left, and we think that may be also due to local movement, maybe diffusive, maybe also due to local structures, which we do not visualize in this kind of microscopy. This is something that we would love to be able to address in future. We built a GUI around it, and this is now a paper, this is a published study in Biophysical Journal. As before, we have also put the code online. Anyone who wants to tweak it, improve it, play with it, you're more than welcome to do so. And so I just want to summarize, and I hope I'm keeping my time, that we've dealt with some simple approaches, some very low-level approaches of gradient-based segmentation of cell shapes for rod-shaped bacteria. Use that to get some insights into cell division and what might be driving it in terms of physicochemical or so-called bulk properties like temperature and nutrient availability. Our length estimates are within a 1 pixel error. And by using a modified Gaussian filtering, what we call scaling of Gaussian, we can enhance the contrast of intracellular particles in DIC images. And when we combine that with single particle tracking, we can actually estimate flows. Those flow measurements seem to match very well with what we have seemingly found already in the in spindle movements. And so we think that now these low level, so there's always I mean, I have a couple of people in my lab, and they think low level image processing is not that great because, of course, there's really beautiful stuff coming out of machine learning approaches. But we think that some of these simple things might also help us plug in to our more complex learning approaches to get more sense of what's going on. So with that, I'm actually going to come to an end. The work I've spoken about has been contributed to by these 2-3 people whose names are highlighted in blue. Dhruv, who is a current PhD student in his 3rd year in my lab. Yash, who was a master's student and is now at the Technical University of Delft, at the Kavli Institute. Anushri, who was a former PhD student in my lab and is now working in medical publishing. The rest of the people have not had an opportunity to talk about their work, but we have a lively group. And most recently, because we've all at least partially been vaccinated, we took the opportunity to explore the surroundings. And so here we are. Funding is, of course, due to the Indo-French Center for Promotion of Advanced Research, the CEFIPRA, IISER Pune, and internal funding, and a CNRS partnership that we have. Marie Delattre has been very kind and generous with advice and thoughts about C. elegans data. So I will stop here and take any questions. Thank you.
Thank you very much. Wonderful. So I think we want to kind of do the traditional first the Mentimeter quiz, which gives you a short break to breathe, and you can comment. Actually, the Mentimeter quiz is a bit of an educational kind of follow-up from what you have presented, and Nick has kindly prepared it and wants to kind of do Mentimeter today. It's his first time, so he's a Mentimeter.
We'll see how it goes. So Tanya, if you stop sharing, please.
Yes, of course.
Thank you.
Yeah.
And everybody who wants to ask questions specifically more in detail, please put those into the chat so we can follow up after.
Here we go. So the link was in the chat just now. So you'll find that in the chat for the seeing of people signing up.
I guess I'll wait off on the questions that are in the chat window. Yes.
Yeah, just wait.
No
you can comment the Mentimeter questions, which you have kindly provided. Once we're there, we need to just wait. People need to log in and.
Yeah, that's just,
yes, no problem.
Can you see that? Well, this is the link up here. Mentimeter. Oh, here we go. We've got some people signing in. Great. Let's wait a bit longer so we can get a few more. Why is it moving to also?
I think we see the link now. Yeah.
All right, come on. Let's see if you get to win a great prize, the Foldscope.
You need to share your screen again, Nick, so we can see the answers.
Yep, sorry.
Otherwise, we just see you, Nick, which, no offense, it's not as interesting as the Mentimeter.
I'm stuck. Just a minute. I can't get back to.
You were almost there. We were almost there. You just kind of. You need to kind of now continue. You just press forward on the bottom of your display. If you put your mouse there, it will allow you to go forward with the slides.
Now, just to clarify, we do this on purpose. When some of us is becoming too proficient with Mentimeter, we change the person that runs Mentimeter.
How about that?
I think you're referring to me. Thank you for becoming too proficient. Yes, okay. There we go.
I think we got the quiz back again. We've got 5 participants. Anybody else wants to sign up? Come on.
That's brilliant. Yeah, thank you. Quite a few good answers there.
Okay. Yeah. Oh, all right. Well, let's just start let's start going.
Yeah, there we go. Now we get more.
All right. Question 1 of 5. Okay.
Come on, guys. We only have 5 players. Yeah, that's good.
All right, we go.
Okay. This may be an easy win, Foldscope. I think this is kind of, you could actually ask to kind of talk it through. Actually, we can.
What we've got.
Right. Would you like me to?
Yes, please.
Do that? Yes.
Okay, yeah. We've got 6 correct answers out of that. Good.
Yeah, I think, Stephanie, you're right. It's a little too easy. The question was really about the basic principle of the DIC imaging. And indeed, most of you got this right with adjusting the orientation of the plane of polarized light being the most straightforward way to do so.
Let's see what the scoring's like. Ah.
Yeah, I think this was testing. Brilliant.
Oh, I think the winner.
So this was the warm-up question, right?
I think I think when someone from your lab is even joining.
I think so. I think maybe they should be disqualified. I can tell you who they are. So it's conflict. They should not be answering.
It's fine. We're happy to accommodate.
No, but they should not be answering. So.
Okay.
Oh, let's see how far they get. We can always.
So, right. So this was about the use of microscopy for C. elegans embryos. And what is the feature that makes it so good for imaging? Perhaps I've already given away the answer already, but is it the genetic tractability? Is it the optical transparency? Is it large size or lack of comparable genetic models?
People are listening to you. Let's see what the scores are. All right. Oh, it's neck and neck.
Let's see. It's not too bad.
Delicious is still winning. Anna's with Lola. Gotta get a bit faster, I think. Okay.
Okay.
Next question.
So I guess the next question is about bacteria and the image formation. What is the nature of that image? Is it shadow-like with a central axis? Is it shadow-like with a central axis in rods but not cocci, heterogeneous shadow, or uniformly illuminated objects?
We've got some quicker answers this time around.
Yeah.
Right.
So this was the trick part because I didn't say anything about cocci. And it's interesting because we tried this. We actually did work with Staphylococcus aureus, and we were not that successful. And that's partly why I thought I might, but if the opportunity came up to talk about it. But either way, it's rod-like that works really well. And this is partly the limitation of this approach. And we need to maybe think of something better and more general. And we would love to do this in future. But so the fourth question is now, okay, the oscillations of the spindle can be seen in DSE through which aspect of the spindle and through which feature? Options are clear region of the poles marking the centrosomes. The central spindle cannot be seen in DSE, it requires a dye, so you need fluorescence label or something. Excluded region, no control where spindle empty fibers are located. Astral microtubules whose lengths increase and decrease.
Wow, there we go. Correct answer. Thank you. You're really having to speak fast there. You're saving me a lot of effort. Thank
you. I'm practicing.
Well, I think the ones which have two possible ones is actually tricky. So that's always kind of difficult. Oh, no. So I think it's someone from your lab. No, Stingalicious. So that's good. Maybe you know what.
Right. I think you got one more shot at winning, I think, is the answer here.
So Dhruv is from my lab, if he is the Dhruv I'm thinking of, but either way.
Well, yeah. I think if he wins, then maybe Nick Barry is going to get a full scope.
All right. All right. The question, oscillations of the spindle poles during anaphase for an approximately 5-minute period are of what nature? In phase, out of phase by half wavelength, out of phase by quarter wavelength, and the phase difference varies. Dhruv, please don't answer.
I think, I'm not sure you gave the answer to this one. Let's see. Oh, yes. Right. Here we go. Oh. We win, I think.
All right. I think it's probably going to be sent then to Nick Barry for.
I think you should get the Foldscope anyway. Just for competing.
No, that's very funny.
At least he was fast on the click on the buzzer. So
I think it would have been embarrassing otherwise, you know. Now there was the pressure to actually win because it had to be. Oh no, come on! Wonderful, that's very nice.
I'm sorry, this was not something I arranged, Stephanie. I apologize.
It's fine. It's very nice.
Nick,
we enjoyed this.
If you could identify yourself in the chat, just so we know where to send it, that would be great.
So I think, Nick, you can stop sharing the screen now, and then we can. Yeah, brilliant. So we had, I don't know if we have more questions now in the chat, but otherwise, I think if people have questions, they can also identify them and just kind of show your Zoom face and talk to us.
Yeah, I see a question, and perhaps if Francesco is around, maybe he could ask.
Oh yeah, that is. Yeah,
that's it. Either way. Yeah.
Yeah. Please pick it up. Yeah.
Yeah. Okay.
So I think we were a little bit distracted by the joy of Mentimetering.
No. I think, yeah, but yeah, I think it was compensated for me by seeing an old friend. Francesco, your question is really nice. So you didn't miss the point. I did not talk about 3D single particle tracking, and part of it is I think for us, part of it is limited by the fact our imaging is really single plane imaging. But if we do get 3D data, then we run into the obvious question of reconstruction of some form, because I fear we may end up oversampling, and I'm not sure it's that great an approach from that perspective, which means we need a better model of those granules. And if we do that, we may get somewhere. This is my take on this, and we haven't done it yet. So maybe I'm sorry, it's not really a good answer, but perhaps that at least is partial. Does that answer the question, Francesco?
Yeah, okay. But actually, I had a question.
Sorry, can you hear me? Oh, yeah, yes.
Yeah.
Thank you very much, yes, Chaitanya. Yes, I cannot open the video, but thank you very much. And it was nice to see you.
Likewise. I guess you're in Frankfurt now, right? I mean.
Yeah, here it is. Yes, I am in Frankfurt. Exactly.
Yeah. Super. Yeah. So, Francesco, would you? I mean, coming back to this, are you aware of a way one could, in a potential sense, get 3D information of these kind of relatively regular structures? Say if we were to even Z-stack them while we're imaging in time, because as of now, we haven't tried it, and I'm not sure it might be that easy, because obviously, as you go, let's say we sample through the plane and we have the profile of a spherical object come up, it's going to change the so-called shadow region. And I'm not sure how one would go about building it up, because I can think of a few things, but to my mind, it might be simpler if we could get a label on it. And that's not easy because these yolk granules are a little tricky to label. So do you have any thoughts on this maybe? Because I mean, I know, yeah, I'm sorry, I'm supposed to answer, but.
Yeah, no, indeed, I'm not sure how it would be possible to track 3D using DIC. I think I have kind of intuition that could be possible, but I don't have any particular specific approach in mind.
Yeah.
Indeed, in the old times at the MBL, we did 3D tracking by using dark field, but of course this is understandable because you can measure the diffraction rings. But this is not immediately possible with DIC. So
correct.
Yeah.
Okay. Actually, I have a question. So you sort of gave examples of two different, I guess, well, very different types of cell. But one was an organism where one might think, when I divide, I basically want to separate myself into two identical copies, or at least share out the materials as equally as possible in the case of bacteria. Whereas obviously in the C. elegans, it's trying to make an asymmetric body out of starting from a single cell. But in the bacterial case, what do you think's going wrong there if it ends up with very sort of non-uniform division? You end up with two different sized daughter cells, for example.
A very nice question, Barry. Thank you. I'm sorry, Nick. So this is a question that troubled us for a while. And from those images that I showed you, we don't get much because obviously they're not time series. But since then, we've been doing a lot more analysis. We looked at the same problem using both the sort of traditional agar pad method, which is that we have a nutrient agar pad on, and we image cells as they grow into microcolonies, as well as using microfluidics where we have control over the cells, where we capture the so-called mother cell, and the other pole is washed down a central nutrient channel. It's the mother machine, as it's called.
Yes.
Right. So in both of these approaches, we found that those cells, I mean, this is a really neat question, but those cells that seem to elongate, when they do eventually occasionally divide, may form mini cell-like structures. We haven't followed their nucleoid, but it looks like they may be devoid of the nucleoid. And they're kind of sick. So sick in the sense that some work we did subsequently in a study where we looked at the replication fork stochasticity, it seemed to suggest that the reason for this kind of unusual elongation is a failure to segregate when the remaining machinery seems to have a be capable of forming the septum and dividing it into two equal halves. As a result of which, we rarely capture, even in these these time series analyses, we rarely capture asymmetric divisions, but simply we either have symmetric division leading to equally sized cells or no division. So I don't know if it answers your question fully, but essentially failure of division is what seems to lead to those filamentous structures.
Yeah. There's a sort of self-correction going on somewhere down the line.
I think so. Right. So this comes back to some of the molecular players that are involved in cell division in E. coli, at least. And this tubulin-like molecule, a cytoskeletal element, which forms this ring, which is then referred to as a Z ring, its assembly is inhibited. In fact, it seems to be the convergence point of multiple biochemical pathways, including the nutrient sensing pathway, and which, of course, then explains the role of nutrient availability in cell size variability. But from our evidence, the reason why temperature or nutrient availability might also influence the heterogeneity in a population might simply be that, or at least we could show using, well, the phosphorylation state of the protein called RECA, which is a recombinase, and the depletion of nucleotides in order to slow down to make replication less processive. So it sort of doesn't continue regularly, but stop stalls. Right. That when we do that, we see higher frequencies of these filamentous cells. And we think this is what probably is driving it. So I think it's a very nice point you made about the contrast between symmetric and asymmetric division, because indeed the first embryonic division of E. coli, of C. elegans is asymmetric, but and that's something that we're looking at a little bit about whether the cytoplasmic motion also reflects this. So this is where we are trying to get more detailed maps of the motion to know whether as we think, the difference in forces in the anterior and posterior regions are reflected also in the granule motion. But for the moment, I think we're still at a slightly earlier stage. So okay. We have not answered your question.
Thank you very much for that. We've got any more questions coming in?
We have actually Daniel Zickers in the audience. He's actually the master of interference microscopy, also looking to measure dry mass, I think, as using the interference, very advanced techniques. I don't know if he wants to. I'm wondering if the difficulty in measuring the different bacteria is actually to do with the dry mass, potentially. Sorry, I'm just a comment, but maybe Daniel is too shy. I don't know if he's known for being shy, but maybe. Oh, there he is.
I'm here. Okay. So, so we did a bit of yeast, right? And sort of mainly growth. We haven't done bacteria. So just in general, I wondered, we see flow of the bulk material, right? So I just wondered whether there might be a difference between movement of vesicles and transport of the bulk material inside cells, which you can see by the interferometry. With the DIC, this is more difficult, and then there is a slow gradient in the change. But obviously the vesicles are very important, as it has been shown.
So if I understood you correctly, you are sort of asking whether we are interrogating, whether there is something else additionally driving these granule motions, and whether we can also say something about the one might call fluid flows. Yes, I mean, is that, did I understand it correctly?
No, I mainly meant in addition to what you see. So I suppose this is mainly the motors. I suppose some might be in the flow. So there is some sort of flow based on contractility in sort of mammalian vertebrate cells. This is what I'm mainly looking at. So it's a slightly different process. I wonder how these two might complement. You know, some of the vesicles might go against the flow, some might go down the flow.
Yeah,
it's just another thing, right?
Right. And I think it's a nice point that you make, because I didn't really elaborate on it, but in some of the work, at least in the C. elegans case, that has been done by Cliff Brangwyn and folks. I think he was formerly in the Netherlands, but they showed that with respect to the granule movement, the flux of granule movement, and I'm just going to pull this up just as a reminder of what I am talking about. The image that you see here of intracellular flows, again with the yolk granules that we also have analyzed, they found that in their particle image velocimetry, they found the net flux in the posterior region and back into the anterior region, the movement of mass is seemingly conserved. There are other studies also in C. elegans that show there is an underlying additional cortical actomyosin flow. And I think perhaps that may be something that may relate to what you were mentioning, because that drives the PAR polarity protein segregation, which in turn then eventually determines perhaps motor or force asymmetries, which then downstream in turn perhaps results in as it appears, results in the asymmetric division. So there are at least two independent processes, and it would not be, they're all immersed in the same medium, so it would not be surprising if they're interacting with each other. That is to say, the microtubule motor system with the astral microtubules contacting the cortex and the cortical flows. And I'm not really sure how they do so. So I may be at risk of sounding a little, maybe I don't want to say more about this, because I think I'm not sure about the answer, whether they do, but I definitely think it might be interesting to look at it.
Yeah. Thank you very much. I think you should have a longer conversation without in later time. So please contact each other. And I need to go now to another meeting, but thank you very much also for pronouncing my name in German, actually, properly. I noticed you said Stephanie, which is the German pronunciation. So thank you very much.
Mean to.
No, that was much better. No, I'm much better than me pronouncing your name, probably. But thank you very much for this. I need to go now. If there's any more questions, then I think I'm sorry, we then would need to kind of close now, but I hope we see you as a person, a real person. We meet again, all of us as a real people, not just in Zoom world. But on the other side, I think it does make the meeting actually possible so easily for all of us. So we are very grateful for you putting yourself forward and bringing this wonderful subject up and showing us really inspirational images and also the discussion. I think everybody should go back to the lab now and do DIC imaging if they haven't done so yet.
I think it's probably time to wrap up anyway. So thank you very much, and
thank you.
Now
wonderful
4-week break, I think.
Hi everyone.
Thanks also for the discussion. Thank you.
Keep well.